The overall goal of this procedure is to determine the extent of microvascular endothelial cell inflammatory activation by quantitating the relative number of neutrophils that adhere to the endothelial cells in vitro under static conditions. This is accomplished by first treating healthy, confluent monolayers of microvascular endothelial cells with the desired experimental stimulus. During the second step, neutrophils from healthy human volunteers are isolated and then labeled with calcium AM. Next, the labeled neutrophils are added to the endothelial cells and allowed to adhere for 20 minutes, during which time a pre-wash measurement is obtained by fluorescence spectrophotometry. In the final step, the non-adherent neutrophils are washed away, after which a post-wash measurement is obtained. Ultimately, the percent and relative adherence of the calcium-labeled neutrophils to the endothelial cell monolayers under each treatment condition can be determined.
This method can help answer key questions in the field of human inflammatory biology, since it can determine the relative extent of microvascular endothelial inflammatory activation under different conditions. In addition, this assay has the potential to be used as a tool to study human endothelial-based disease processes that involve leukocyte binding to the microvascular endothelium.
On the day of the assay, use phase contrast microscopy to confirm that the cells exhibit a healthy cobblestone appearance with little cell debris.